Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I'm going to look at your fish exchange surface. Now, if we were to look at our bony fish, our large bony fish, things like trout, cod, we know that they are very active and they have a high oxygen demand. They require aerobic respiration for the ATP so that they can move around and be as active as they need to be. Now, their scales, which are on the surface of their body, does not allow gas exchange. Diffusion in water is much slower, and their surface area to volume ratio means that diffusion alone would not supply enough oxygen to the cells inside their body. So bony fish have had to advance their ventilation system so that they are able to overcome these challenges. And what we're going to look at today is exactly how they have adapted their respiratory system and therefore these exchange surfaces. So the first thing we need to look at when we talk about the fish exchange surface is what happens inside the mouth of the fish. So we can see here that the fish can open and close their mouth. Now, by opening the mouth, what's going to happen is some water will actually move into the mouth, but that's not enough of a movement. So what needs to happen is when the mouth opens, what the fish will then do is they will lower what's called their buccal cavity. Now, by changing the size of their buccal cavity, they are increasing the volume. And when they increase the volume inside their buccal cavity, that is therefore going to decrease the pressure, and that will allow for water to move into their mouth. Now, if they are a cartilaginous fish, then they don't have the ability to do this, and therefore they do what's known as ram ventilation. So they basically ram the water into their mouths. Whereas these fish, the bony fish, they don't continuously swim. They have times in which they stop. So by adapting this ventilation system, it means that they are able to maximize their exchange surface and maximize the amount of water that's flowing into the mouth. So the mouth is open, the buccal cavity is going to lower, that is going to increase the volume, decrease the pressure, and water will move into the mouth. Now, if we look from a top view then, what we can see is our mouth is open, our buccal cavity has lowered and therefore created a greater volume inside the buccal cavity. Now, what's important for us to know is there's also what's known as the operculum. Now, the operculum is the bony flap which covers the gills, and inside the cavity is called the opercular cavity. So the buccal cavity is before the gills, the opercular cavity is after the gills. So by increasing the buccal cavity and having the mouth open, that is going to cause an influx of water into that buccal cavity. Now, what the fish will then do is they will actually expand the opercular cavity. By expanding the opercular cavity, they are changing again the volume. The floor of the buccal cavity is going to move up, and what's going to happen is that's going to again increase the pressure, and that will cause water to move over the gills and into the opercular cavity. Once the mouth is then closed, what will then happen is the opercular valve will open. And after the opercular valve has opened, the sides of the operculum are going to move inwards, and that again is going to increase the pressure, which forces water out of the operculum. The buccal cavity is going to constrict, moving upwards more, and that will maintain that flow of water over the gills. So the water is moving in, the water is moving into the buccal cavity, across the gills and out of the operculum. So it's important that we note, why do fish do this? Why is this the best way to ensure that there is exchange of gases occurring. So to understand that, we have to really focus in on the gills, and we need to look at what's known as the gill arch. So the gills are made up of a load of gill arches. Now, gill arches has 2 rows of gill filaments to them. So each of these gill filaments are made up of many lamellae, and those lamellae increase the surface area. Now, what you have is that the gill arches lie where the tips are actually touching each other, and so therefore water needs to flow between the gill arches, and therefore maximizes that surface area that is exposed to the flow of water. Now, by having the gill filaments then consisting of these lamellae, what we have is that water is going to flow between each of the lamellae, again increasing the surface area. So again, we are maximizing this exchange surface. Now, those gill filaments, which consist of those many lamellae, actually have a really good blood supply. As you can see here, there is oxygen poor blood moving in and oxygen rich blood which will flow out. Now, the water flows in one direction and the blood will flow in the opposite direction. Now, that is known as a countercurrent direction. Now, that countercurrent direction is needed to maintain a concentration gradient. So if we have a look at the percentage saturation of oxygen in the blood compared to water all the way along the gill plate, what we will see is that the water concentration is always greater than the blood the further along we go through the gill plate. That ensures that the oxygen is always going to diffuse from the water into the blood. Now, the fact that the gill plates are actually touching at the end, the fact that they're so close together, slows the flow of the water down across the gill arches and therefore maximizes that efficiency of that exchange. So you also need to be able to discuss how you would do a fish head dissection and what is needed to ensure that you can see the different parts, the operculum as well as the gills. So the first thing you would do if you're going to do a fish dissection is actually take a set of forceps and push them through the mouth and out through the operculum. What that will do is that will show you the flow of water going into the mouth and out of the operculum. What you then want to do is actually lift up the operculum and be able to see the gills below. Now, you then want to remove the operculum with a pair of scissors. So by removing the operculum, that will allow you to observe the gills more clearly. Now, what you would do then is using either scissors or a scalpel is you want to remove the gills. Now, remember, we're talking about bony fish. So to be able to do this, you will need to cut through the bone at the top and at the bottom of the gills, and that therefore will remove it from where it's attached to the head. Now, by placing the gills in a beaker of water, that should allow them to fan out, and that will allow you to observe those filaments more clearly. Then once you have observed them, you will either make a biological drawing with measurements, or you may actually try and look at them down a microscope depending on what your school does. So if you like this video, then please do click the like button and subscribe to my channel. Also check out my revision platform www.aiqchat.com if you want some more help with your revision.